So today we're going to dive into the details of the light reactions. And so remember from last time where this is happening. This is happening inside the chloroplast. If we're talking about plants, this is happening in the chloroplast. And specifically, it's happening in the thylakoid membrane. And so on one side of the membrane, we have the stroma fluid that's inside the chloroplast, and on the other side of this membrane is the lumen of the thylakoid. And we had talked about last time that we have arranged in these thylakoid membranes photosystems that are absorbing light, and they're doing that using pigments, photopigments, in certain arrangement with proteins. And so these are forming photosystems, and the photosystems are absorbing light in the thylakoid membrane. So the green blobs here that start with P, those are photosystems. And what we're going to be looking at here is these photosystems are included as part of an electron transport chain. So this is a little bit similar to mitochondria in that there is an electron transport chain moving electrons down the line within a membrane. But the difference is here that we have light is coming in and exciting electrons and getting them to jump up in energy. So we're starting with electrons excited by light instead of starting with electrons from high energy chemical bonds like we do in aerobic respiration. So there are two different photosystems involved here, and they are named photosystem 2 or PS2. So you can also call this PS2. It's just as exciting as a PlayStation. And this one is PS1. Why are they named that way?
That's the order they were discovered.
Yes. So the first one that was discovered and isolated and worked out was photosystem 1. And then after further study, it was discovered, oh wait, there's another one that actually occurs earlier, and that one was named photosystem 2. So that's a thing to memorize. Their numbers do not correspond with the order that they're moving electrons. Okay, so this is kind of an overview slide, and this would be one of your key figures to study light reactions. So I'll be coming back to this picture pretty soon. All right, so we're first going to look at photosystem 2. This is the one that is occurring first during the light reactions. Get this pulled down exactly where I want. So it's absorbing light. There are complexes of chlorophylls and proteins embedded in the thylakoid membrane. And this particular photosystem, its maximal or its best light absorption wavelength is 680 nanometers. So this can be called photosystem 2 or P680. Either way, and either designation is okay with me. So its best absorbed wavelength of light is 680 nanometers. Yeah, I've got that on there. So light is being absorbed. Electrons are jumping up in energy and being passed on to a molecule called plastoquinone. So when the electron gets excited enough, gets to a high enough energy level, it can reduce a molecule called plastoquinone. And those of you that are in cell, notice that this is sounding kind of like ubiquinone. They're chemically similar, but this one is in a chloroplast and it's called plastoquinone. It's going to be somewhat challenging to not mix those up, but just don't mix those up if you're in plant and cell at the same time. Plastoquinone is going to pick up that energized electron. Plastoquinone is also going to be picking up a couple of protons as it reduces plastoquinone. So again, we've got protons following along, kind of like groupies following the electrons. So when that happens, we had an electron that left photosystem 2. Two of them, actually, that left photosystem 2. They have exited the building. And if that process is going to keep happening over and over, those electrons need to be resupplied. And in fact, they are resupplied by photosystem 2 also being able to split water. So part of the energy that is absorbed by this photosystem goes into boosting electron, and some of the energy also goes into splitting water into molecular oxygen, protons, and also electrons. It says half O2 because there are multiple waters quickly going through this process. Light is boosting electrons, and light is also splitting water to resupply electrons that just left the photosystem. So this place right here, this is the place that the plant needs water to do photosynthesis. This is where water is involved. And water in photosynthesis is a source of replacement electrons, or to resupply electrons. What's oxygen doing in this? Is it doing anything? What's its purpose here?
Does it donate the electrons?
Does it donate the electrons? Water donates electrons. Oxygen is left over. Do plants need oxygen? They do in their mitochondria, but they don't need oxygen for photosynthesis directly. So here's some oxygen. It's made during photosynthesis, and therefore we say that this is a type of oxygenic photosynthesis. It makes oxygen. It does not use oxygen, it makes oxygen. So where's this happening? Where are plants doing this?
Thylakoid membrane.
Thylakoid membrane, inside the chloroplast. And remember that the chloroplast itself is derived from bacteria. Chloroplast is derived from bacteria. The chloroplast is doing oxygenic photosynthesis. So this process of making oxygen is older than plants. Old, old, old. This is how we got oxygen on the earth. Remember that first section of class when they said there was oxygenic photosynthesis, and it reacted with iron to form banded iron, and then we had ozone, and then we had oxygen building up in the atmosphere. That's older than plants. This process is older than plants. And this oxygen that is produced, the plants do end up using it in their mitochondria when they do aerobic respiration, but they didn't come up with that. They're not using it in photosynthesis. It's a byproduct. They use it in aerobic respiration, which they also do. Is that making sense? I'll come back to that later. So this is where water gets used. If you go back to that summary figure, the best study figure, not this, not this, not this. Where is it? This one. This one's my favorite. So we've got light reactions light's getting absorbed, water's getting split, oxygen's being produced. That fact about where's oxygen involved, where does water get split, that comes up a lot in standardized tests. Not just my tests, but that's a thing that standardized tests like to ask. Where does oxygen get produced during photosynthesis? It's in the light reactions. Okay, my next question is, where does oxygen go now? What happens to oxygen after this?
Can you go into the cytosol?
Yeah, it goes everywhere. It diffuses away from here. So it goes into the stroma, it diffuses into the cytosol, it diffuses out into the airspace around the plant mesophyll, and it goes into the air and we breathe it. So it's diffusing out of there. That's going to become important when we talk about other mechanisms of alternative photosynthesis. So the next thing I want to talk about is down here. Water's getting split. It's producing oxygen gas, it's producing protons, it's producing electrons. And that process is also generating free radicals. And we've got electrons moving generally away from the partners. So we've got reactive oxygen species being generated or free radicals. And those are really damaging if they float around in the plant and react with other things. So we want to have electrons going this way, free electrons going into the photosystem, not bouncing around out here. And manganese is really important for that. So we already saw that magnesium is really, really important plant nutrient because it builds chlorophyll. Manganese, Mn, that we don't talk about much in other aspects of biology, it's really important as part of the photosystems. So it's an essential plant nutrient. And the manganese atoms are able to hold unpaired electrons temporarily. So it's kind of like a holding cell for, I don't know, dangerous criminals. Like, no, don't be walking around out there. Stay here. Stay here until we can get you to the right location, and then you won't cause any more damage. So manganese atoms are acting to hold on to the electrons and help them move up toward the reaction center to do replacement for the ones that left. So if you look on plant fertilizer, it always has manganese for this reason. It's critical for the photosystem. So that's generally what's happening with photosystem 2. So the electrons are excited by light, they're resupplied by splitting water, they're passed on to plastoquinone, PQ, along with some protons. And notice here that the protons, where those protons are coming from. The protons that are being picked up, they're coming from the stroma side. It's very intentional that the protons that are being passed on to plastoquinone, they're coming from this side. So once plastoquinone has the electrons and it has the protons, it's reduced. It's holding electrons. And it floats, or it diffuses, within the membrane. It never leaves the thylakoid membrane. It's hydrophobic, and it can drift laterally, but it doesn't leave the membrane. And it will donate electrons onto the second component, which is cytochrome B6F. This is a term that you should memorize. Cytochrome B6F. And when it drifts over near cytochrome B6F, it donates the electrons where it reduces cytochrome B6F. And it also drops off the protons. And notice where it drops off protons. The protons that came from the stroma side are now delivered to the lumen side. It's like if a car picked me up on one side of the street and it dropped me off on the other side of the street. I've crossed the street now. I'm on the other side. So this has moved protons from one side to the other. It's not pumped protons exactly, but it's had the effect of moved protons from here to here. And by doing that, we've acidified inside the lumen, because there's more protons in here now, there's more acid. And we've made down here also a proton gradient. There's now more protons inside here than outside, as long as this continues to operate. And this is also kind of similar in concept to what mitochondria do. Except in mitochondria, we're putting protons into the intermembrane space. It's flip-flopped. But we've got a proton gradient in here. And the only way that they can come back out is through ATP synthase. This is the only exit for protons. We'll come to that in a minute. Questions so far? Looking at how many of you are in cell. You might want to study these things side by side, not to mix them up. So at this point, we've reduced cytochrome B6F. We've moved some protons into the thylakoid lumen. And the next place that the electrons are going to go are onto this PC, which stands for plastocyanin. Plastocyanin. And this is sort of serving a similar function to cytochrome in the mitochondrial electron transport chain. But again, it's called plasto. It's in the chloroplast, plastocyanin. And it picks up electrons from cytochrome B6F and moves them over to photosystem 1. So let's look at where electrons have gone so far. Electrons have been excited by light, passed to plastoquinone, passed to cytochrome B6F, passed on to plastocyanin. And the ones that left here are replaced by electrons that come off of water. Water's for replacement electrons. So now they're on plastocyanin. And plastocyanin are going to deliver electrons to photosystem 1. Photosystem 1 is operating generally the same way as photosystem 2, but it's got a slightly different wavelength preference. It absorbs best at 700. So you can call it PS1, you can call it P700. Either one are okay with me. And the electrons, when they get here, they have lost some energy. And this box should not be on here. This really goes on the next slide. So just ignore that on this box. So the electrons, once they arrive here, they have lost some of their energy along the way. Every time they get passed on to the next one in the chain, they've lost a little bit of energy. And the electrons, by the time they get to photosystem 1, they have less energy than they did, but light strikes again, and light boosts these electrons up again. So when they arrive, they're a little bit tired, low energy. They get a light boost. They gain energy from that. And they're going to end up leaving photosystem 1 on the stroma side through some iron sulfur proteins there. Once they leave photosystem 1 with their new energy boost from light absorption, they're passed on to a molecule called ferredoxin. Ferredoxin, so like associated with iron, ferredoxin. And ferredoxin, before it has the extra electrons, it's oxidized. When it gains them, it's reduced. So it's picking them up on the stroma side. So it's picking up electrons on the stroma side from photosystem 1. And it's going to carry the electrons to an enzyme called ferredoxin NADP reductase. And that enzyme tells you what it does. So it picks up electrons from ferredoxin and uses them to reduce NADP. It's putting electrons from ferredoxin onto NADP, forming a new bond, a hydrogen jumps on too, and then we get NADPH. And NADPH is one of the major things that plants are interested in making out of the light reactions. So on your quiz, that would be one product of the light reactions, NADPH. And remember, this is reducing power. And NADPH can deliver electrons elsewhere in the plant into new covalent bonds. NADPH is one of the major things to come out of the light reactions. The other thing about this process is that getting electrons energetic enough to add on to NADP+, this process requires the electrons to be really high energy. They have to have sufficient reducing potential or reduction potential to be able to do that. It's kind of like saying NADP has really high standards for what electrons it will accept. They've got to be super energized to be able to participate in that bond or to form NADPH. Last thing I want you to notice about photosystem 1 is where photosystem 2 is dependent on manganese. Photosystem 1 is very dependent on iron and sulfur. And again, if photosystem 2 is using manganese to pick up free electrons and hold on to them temporarily, a similar concept applies. Photosystem 2 is getting ready to pass electrons into a system, and we don't want them to get out of control, bouncing around, causing oxidative, reductive damage. So iron sulfur proteins are sort of, now hang on, don't leave until we're ready for you to leave, hanging on to those electrons right before they get passed into the stroma. Okay, so that's how we make NADPH. So far in the light reactions, we've made oxygen as a fortunate byproduct because we split water. We've made NADPH which is important as reducing power. And we haven't talked about ATP yet. That's where we're going next. So if I skip back a couple of slides to our overview, electrons have come from water in general, and then they've had an odyssey onto NADPH. That's the path of the electrons. While they were on this path, there was a proton gradient formed too, and that's what we're going to look at next. So, all right, we've got a lot of protons in here now. We've got potential energy by them being packed into the lumen, and they can exit through ATP synthase. They're going to exit through ATP synthase, and it's going to spin, and it's going to use that spinning power to join together ADP and free phosphate to make ATP. This ATP synthase is working generally the same as ATP synthase in mitochondria. They have slightly different shapes, but overall they're doing the same thing. They're using a proton gradient flow through to make ATP, and they do that by the method of oxidative phosphorylation. And that's where the ATP comes from.
So after the hydrogen goes through the ATP synthase, does it just hang out there and wait to start over again at the very
Joins
beginning?
the pool.
Yeah, okay.
Yeah. So this particular one, if we wanted to track one, it would be diffusing around in here. It could eventually make its way over to another photosystem too. That reminds me, something else really important about this, and I will also be saying this tomorrow in Cell as well. When you learn about these electron transport chains, you learn them in order. It's like, well, first there is photosystem 2, and then it's next to a plastoquinone, and then it's next to a cytochrome B6F, and they're in this line. They are not really in a line. They're not in a line. The book shows them in a line. If you imagine the thylakoid membrane as a floor, like if we were to look down on it in the floor, these would be scattered throughout. These would be scattered throughout. So would these. They are not in lines. What keeps them in line? How do they know who to donate electrons to?
Does that have to do with the energy level of the electrons?
The reduction potentials, yes. Or the energy level. Like, I only give out electrons that have one light boost worth of energy, to make it really simple. I only take electrons that have one light boost worth of energy. I only take electrons that have the maximum energy available after 2 light boosts. That's what keeps them in line. They're not in the line. They're mixing around in the membrane. And that's true for mitochondria as well. They're not in the line. But they donate, so they're not in the line, but these components donate and receive electrons of specific reduction potential. But I still, remember them in the line, even though they're not. That's how I remember them. All right, so here's our cartoon of ATP synthase. And maybe this is the first time that you've seen it in this configuration where it looks like it's upside down. There's no up and down. There's one side of the membrane and the other. And so for plants, if you think about where are the protons building up? Where do protons build up in plants? In chloroplasts, let's say. Where's the acidic place?
Inside the cord.
In the lumen. And that makes sense because the lumen is kind of keeping them sequestered away from other places where that much acidity might be damaging. So they're building up in the lumen of the thylakoid in a plant. Where do protons build up in a mitochondrion? In a mitochondrion? The intermembrane space, yeah. Which is connected with the cytosol. We'll talk about that tomorrow. That's coming tomorrow. I don't do this on purpose, I swear, but it just happens to. Like you got light reactions one day and proton pumping in mitochondria the next. So protons enter ATP synthase on the thylakoid lumen side and they leave on the stroma side. And thus it looks like now a hot air balloon. In mitochondria, they usually show it in the other orientation where this part's hanging down. It's just one side of the membrane or the other. So I put a question here. Where do the protons for the gradient actually come from?
Water.
Yeah, they do. So let's go back. Some of the protons are pumped over. This is cool to me too. Some of the protons are moved over, and some of them come from splitting water. Both of those are helping to form the gradient. Like double acidity in here. And the only way that they can get back out is to run through ATP synthase, spin it, which helps form ATP. And ATP is a good energy source for the plant cell. This ATP that is made during photosynthesis is mostly going to be used for carbon fixation. So the plant's making ATP during photosynthesis. It's fixing carbon, using that ATP as an energy source. And then this is an important point. Plants then used fixed carbon to supply ATP. So they are having their own cycle. They're making sugars during photosynthesis, and then they burn sugar for energy in parts of the plant that can't do photosynthesis. So what I'm writing here is plants are using fixed carbon that they're making during photosynthesis to supply ATP to non-photosynthetic tissues. So if we look at our box elder out here, it's still photosynthesizing. It's October 9th. It's still green and the leaves are photosynthesizing and they're absorbing light right now, even on a cloudy day, they're doing this. And then the light reactions, they're making NADPH, they're making ATP, they're making oxygen. Still doing it. And the sugars that they make, they'll pump down to the roots. They'll pump into the trunk. They'll pump into any tissue that can't photosynthesize on its own. So they make sugar and they burn sugar. They do make their own food. They still need food. They're just making it on their own. All right. Does seem okay? I think what's hard about this is memorizing the order and the names and knowing what gets produced when. So this picture is really helpful for light reactions specifically. And then our big overview picture, what is it? This one. So up till now, we've made it to here. We've made ATP, we've made NADPH. Oxygen has been produced as a byproduct. And remember, these are now floating in the stroma. The ATP is in the stroma, the NADPH is in the stroma at the end of the light reactions. There we go. All right, the next thing that I want to look at is, we're looking at that same process, but in a different view. So this was showing us, all right, if we were to look sideways through the thylakoid membrane, that's how electrons are getting passed. This view is looking at, yes, electrons are getting passed one to another, but arranging them by their reduction potential. The higher the electron is on this graph, this is really a kind of graph, the more energy it has and the better reductant it is, the greater its reduction potential. So the electrons that are split off of water that do replacement, they're actually not that high energy level yet. How do they get up to this energy level?
Light.
It's the light energy. Gets them moving faster, gets them to jump up in energy level. And so when it's saying P680 like this, P680 like this, that's got high energy electrons. It's been excited by light. They're at a high reduction potential. But they actually are not quite at the level of reduction potential to make it onto ferredoxin. I mean, it looks like it's close on this graph, but these electrons at this level are not good enough yet to reduce ferredoxin. And ferredoxin is what passes electrons onto NADP reductase. They've got to get double boosted to be that good to make NADPH. So here's the light boost. That gets them up to this level. Then they're good enough to reduce plastoquinone. And after they've ridden on plastoquinone, they can reduce cytochrome B6F, reduce plastocyanin, fall into photosystem 1. And the second light boost gets them at a high enough reduction potential that they can end up helping to form NADPH. This is called a Z scheme. Z scheme because if you look at it sideways, it's kind of silly. It looks like a Z. So I want you to associate Z scheme. Plants do a Z scheme in electron boosts as part of their photosynthesis. Cyanobacteria also do. So the bacteria that do oxygenic photosynthesis, they do a Z scheme. There are other organisms, mainly bacteria, that do photosynthesis that do single boost. It's called cyclic electron flow. They boost up, they reduce something which is not to this level. The electron goes right back to the photosystem, and it goes through cyclic electron flow. Z scheme is special to plants and cyanobacteria, and you should associate that word with light reactions. It's a way to get electrons boosted high enough to have fantastic reducing potential. Cyanobacteria kind of invented that system, and then plants got it too when cyanobacteria developed into chloroplasts over the years. Oh, here's a picture of a non-Z schemer that still does photosynthesis. So there are bacteria that do photosynthesis, but they're only doing generally half of what a plant does. So boost the electron, reduce something, use its energy, pass it back to the photosystem to do cyclic electron flow. You might add on to here also, cyanobacteria do this as well. So here in our review, this contains in it like all the things that you need to know about the light reactions, except for possibly the compartments. What's this compartment? This is a stroma. What's this compartment? The lumen. I'm going to call it thylakoid lumen. And what's this membrane? Thylakoid membrane. And we can look on this and see what are the major products. This is something that you should memorize as well. So the major products of the light reactions, we've got oxygen, we've got NADPH, and we've got ATP. Remember, these two are now available in the stroma, and the stroma is where the Calvin cycle is going to take place. So that's convenient. They're already there. They're available for where they're going to be used next. Oxygen diffuses through the chloroplast, through the cytoplasm, and across the plasma membrane into the air space. Oxygen diffuses away. It will diffuse through all the liquids in the chloroplast, all the liquids in the cell, and when it gets to the boundary of the cell with the air, it also diffuses out into the air. And then it goes out the stomates. And then it goes into the outside air. It goes everywhere except across the cuticle. Will diffuse out. So we're going to see, maybe Friday or maybe next week, that if plants close their stomates, the oxygen gets held up inside the leaf. And that can actually cause problems for the plants in certain conditions. So it's not staying in the lumen, it's diffusing away. Okay, if someone would turn that off, that would be the end of our